Hello, my name is Gabriella Scarlatti. I'm the Science Director of the Global HIV Vaccine Enterprise. The Enterprise is an alliance of independent organizations that collaborate to accelerate the development of safe and effective HIV vaccines. We mobilize stakeholders, we identify collaborative opportunities, and we promote sharing of knowledge. In specific, one of our programs is e-access meetings. E-access meetings serves to disseminate research findings that were presented at meetings so that it reaches a broader audience. Today it's a pleasure for me to moderate a panel discussion on HIV vaccine path to efficacy. This is a joint initiative with Keystone Symposia, which convenes some of the leading life science conferences worldwide. Let me introduce our panel of experts. Nicole Fram, from Fred Hutchinson Cancer Research Center in USA, Barton Haynes, Duke University Medical Center in the USA, and from South Africa, Penny Moore, National Institute for Communicable Diseases. Thank you again for being part of this discussion panel. I would love to start by asking you to introduce yourself and say which is your main area of interest and expertise. Please.
All right. My name is Nicole Fram, and I work at the HIV Vaccine Trials Network. So my main area of interest is HIV vaccines, specifically preventive HIV vaccines. I work in the cellular laboratory, so I'm much more interested in T-cell responses than these guys over here. And I'm looking forward to the session.
My name is Penny Moore. I work in South Africa at the National Institute for Communicable Diseases, and I've been studying virus-antibody co-evolution in HIV-infected people.
My name is Bart Haynes, and I work at Duke University, and I work on HIV vaccine development in general, and in particular on how to create viable vaccine candidates that will induce protective immunity.
So thank you very much. We have an excellent panel here, spreading through from T-cell to B and vaccine research, but you are all involved in the same field, HIV vaccine. So let's start by asking you, HIV research is a rapidly evolving field. There is every day something new coming up. And so I would love to know from you, let's start with you, Bart, what is really the most recent discovery that you think is exciting and that we should all know about?
Well, I think one of the most exciting things that's happened very recently has been the advances made in structural biology and understanding what the trimer of the HIV envelope looks like. We've really wondered about this for quite a long time. And I think the new studies that are beginning are first understand what it looks like and how it holds together, and then what it does when you inject it into animals and hopefully eventually into humans. And I think this is really very exciting. I think the field is really working on the fundamental question of, does it matter what type of immunogen goes into a vaccine, if it has the right spots on it, that are the right epitopes that are needed for the immune system to see? Is that all that's needed? Or does the whole, the context in which it's introduced matter? And so I think we're really making progress in solving that question.
I could go on immediately with a question, because this brings us to the point, do we want really, what type of response do we want really to induce with a vaccine? We have an immunogen that we have new immunogens coming up now. You said it, but which one do we want to use? How do we want to use it? And which is the response that we really want to
I think that part of the excitement about the trimer is that all the targets are theoretically present on a trimer, and I think that's part of why the potential is that you can elicit a polyclonal response to many targets. So I think that's one of the attractions of using a trimer as an immunogen, as opposed to what we've been doing for many years, which is to focus on small bits that normally include only one or two of those targets. So I think ideally we all want a polyclonal response that targets as many of the vulnerabilities on the virus as possible.
Yeah, and just to add to that, I think it has to be polyclonal. That's a very big thing, and just focusing on V2 is not necessarily going to cut it. But it's also going to be important to get the adequate T cell help, just to throw the T cells in there. So I think that from everything that we talk about, I mean, we talk about antibodies, and especially at this meeting, it's been talking about antibodies a lot. But I think that we shouldn't forget that there are other arms of the immune system, whether it's CD4 T cell help to really help induction of proper antibodies with TFH or anything else. But also just as not forgetting about a second line of defense, that if people do break through, it would be so helpful to have CD8 T cell responses are ready to go, to at least reduce viral load in the people that do get infected. So I like the polyclonal as a start for, yes, we need to go beyond just a single thing, but also just broadening it to not just antibodies.
Yeah. I think the other exciting thing that's an emerging theme, I guess, is the germline targeting immunogens, which is, in a sense, the complete opposite of what we've just been talking about. So that's, well, for many years we've known that eliciting these antibodies is very hard, and that there's something special about how these broadly neutralizing antibodies, how they get started. And partly because of structural biology, we now have a better sense of how to engage the precursors and get them going, and then maybe to prime with those and then boost, and actually be able to pick up those very rare precursors that we so far have been missing and push them in the right direction and hopefully drive them. And for me, that's been one of the exciting themes that have come out.
How would you do it?
How would I do it?
Yeah.
How would I do it in humans?
Yeah.
I mean, that's the million-dollar question. But I think up to now, this idea of targeting germlines has been, for me, an optimistic, maybe slightly crazy idea. And I think we're beginning to see some data, at least in mice, with big caveats, that maybe there is some reason to believe that it might work. And I think the next thing is to take those into humans and to prime. And then, as we saw at this meeting, there is the potential to systematically shepherd the immune response along the right pathway. I think it's early days, but I think we're starting to see some encouraging data that suggests it's not pie in the sky, which I think some of us thought it might be for a while.
Well, along those lines, one of the most exciting things that I've heard recently that sort of validates this approach is the work of George Shaw on developing chimeric simian human immunodeficiency viruses. And particularly exciting to what the point Penny just made is that when George took one of these transmitted founder shivs and infected monkeys, in the monkey, this particular virus underwent the same evolutionary pathway that the virus went in the original individual from whom that virus envelope came that was used to make the virus. And while this is extraordinary in and of itself, it really gets to the point that we've all been struggling with. Can you look at antibody-virus coevolution in one individual from one particular country that made a neutralizing antibody response and develop immunogens from what you learn from that individual that would be generalizable to others? And here's an example of where the virus evolution transferred across phylogeny, not just from individual to individual, and was the same in the rhesus macaques as it was in humans. And so that's a real boost for the theoretical approach that Penny is just talking about.
Yeah, I agree. I mean, I think it's an incredible result, actually. I think, you can literally follow exactly the same patterns in the two. And I think that's encouraging. But I think the other thing that's encouraging is now that we've got more and more people who develop these broadly neutralizing antibodies and we study how they've evolved in all those cases, there are parallels. So if you look, for example, at all the V1, V2 antibodies, so V1, V2 is just one target at the top of the envelope trimer that is a focus for vaccine design. If you look at how those antibodies evolved, they've evolved amongst using very similar pathways. And that's encouraging if we think that maybe that means it might be amenable to a common immunogen design that can harness the fact that these have predefined and very similar pathways of development.
So would you suggest also that we could use one single immunogen all over the world?
Well, I think that what we're saying is not that it's going to be a single immunogen, right? You have the germline target, and then you follow up with these designed boosts that actually will differ over time. And so I think that we can make one vaccine that will work all over the world, but it's not going to be one product. So it's going to be different. It's going to be a suite of different proteins, or however you want to package them, to really drive the immune response in that direction. But I think that from everything we know right now, and especially with a jump from human to monkey, or probably the other way around, that it's very promising that, yes, we can actually make one vaccine that's going to be possibly targeting everything. And with the caveat that, of course, clade is going to be somewhat important. But I think that's one of the other things that's very exciting right now is we have all these mosaic immunogens. So if you're able to engage the germline with a proper germline target, and then drive the immune response with mosaics that actually give you cross-clade reactivity, which is, I guess, what we're after anyways with the broadly neutralizing antibodies, I think it's very promising.
And that in and of itself, I think, is really a new observation that the field has come a long way from the early days where the field tried every strategy for every successful vaccine and it failed. And only really until recently, I think the field was really still hoping that a very simple solution to the HIV vaccine problem was going to be found. And I think it's a general feeling now, or belief, that, and it's okay, that no, this is really a very complicated pathogen. It inserts into the genome. It changes very rapidly. The mutation rate is confounding, and the reason for the mosaic vaccines that Nicole mentioned. And so simply acknowledging that this is going to be a complex vaccine, and it may require immunization over a longer period of time than any other vaccine. It's certainly requiring a greater study of how the host pathogen interaction works in order to understand that. And I think if I had to say one sentence, I think the field has understood we have to really understand the fundamentals of the host pathogen interaction to make this vaccine. And accepting that is, I think, is really a step forward for the field.
It brings me to several questions that come up from your discussion. The first one is, how do we go from the animal model to a human? Do we have a real good animal model that we can, for vaccine research? And which one would you choose? And how, is there any go, no-go criteria for your vaccine to move into the human from your animal model?
Well, I think animal models are extremely helpful, and the animal models, but not as go, no-go criteria. And the short answer is because this virus has developed on certainly the primate backbone or the primate immune system and learned to thwart it. And we're having to really evaluate the vaccine candidates in the context of the human immune system, whether that human immune system is in the context of humanized mice. And certainly the field has worked very hard to learn to do almost everything we can do in humans in rhesus macaques, but macaques still aren't humans, and we don't have the perfect challenge model in macaques. So to have that as a bottleneck or a go no go criteria for going into humans, I think it should be more parallel rather than a go no go criteria. I think what we've realized as a field is that we need to figure out how to get more materials made good manufacturing practice and therefore into small iterative phase 1 clinical trials to do the immunology in the context of the human clinical trials in order to learn which one starts the germline off, which one then takes it to the next step and the next step and the next step, and to do this iteratively and to figure out how to do this without each step taking 3, 4, 5, 6 years. We have to learn together how to do this in a shorter period of time so we can bring more to the HVTN and get the answers sooner.
But I think, yes, I absolutely agree. It should never be a go-no-go decision. But I like the idea of iterating not only within humans, but going back and forth between humans and monkeys, because there are certain things we can't do in our trials, right? So doing them in parallel, seeing where do they actually behave comparatively, and then using those models to then better understand, for example, the mucosal immune response, or where does the virus go in lymph nodes, all these things that are really, really hard to do in humans. But I think that it's very hard to convince the funders that this is the right thing to do. So it feels like they still see this as we go into monkeys first, and then we go into humans, and then we're done. And then it didn't work in humans, and we don't know why. And it may still be a promising approach that if we then kind of turn back and figure out where did it go wrong, we could actually move forward in a much faster way.
Well, I think there are a lot of progress is being made in both, and I see what's happening is that we're putting immunogens at the same time into small animals, into humanized mice models, which may well turn out to be quite helpful. And then in macaque models, but not to shy away from taking the most promising immunogens on into humans to get, because we continually get surprises there.
Yes, indeed.
You have some opinion about that?
I guess the bottleneck so far has been the number of things that have gone into humans, and I think that's probably one of the big changes we're going to see in the next 2 or 3 years, is more and more, hopefully, more and more products going into humans. And probably at this stage, we don't have enough products that have been both into humans and animal models to actually be able to effectively evaluate the animal models at all. And I think that's something that might change in the next couple of years as more and more goes into humans.
Absolutely.
I think is a key step for the field.
So the other question I have is a bit provocative, but RV144 showed a lot of correlates. RV144 is the vaccine trial that was completed already several years ago and showed for the first time protective effect. Was good, 30% more or less. But the correlates, none of them was broad neutralizing antibodies. So, do we need broad neutralizing antibodies to have a protective vaccine, or do we think we can have a combination of antibody responses with different functions? What do we want really?
We want everything.
Everything.
I was like, is that a question?
That's not a fair.
We want everything. I think that if we can get broadly neutralizing antibodies, I think we all would agree that, yes, that would be the ideal situation. I think that while we are developing all these new immunogens and these trimers and everything, it's still going to be a couple of years until we have data in humans to show whether or not these are inducing the kinds of immune responses that we want. And I think that we shouldn't just neglect other functions of antibodies that might provide to be, at least help, even just in the context of neutralizing antibodies, might help the immune system and might provide, again, another leg for it to stand on and to prevent infection. So I'm very much in favor of maybe even focusing the research on the induction of broadly neutralizing antibodies, but I think that we should really not forget about everything else. And there are trials right now that are going to be starting in South Africa relatively soon that are built on RV144 that are hopefully drive protection a little bit further if all goes well, and they won't be inducing neutralizing antibodies. I think we're pretty sure of that, or they'll be weak tier 1 neutralizing antibodies that really are not what we're looking for in general in HIV vaccine research. But I wouldn't dismiss it. Again, I think that the broad, and that's, again, comes to the polyclonal response that Penny mentioned before, the broader we can get, the better we might be able to prevent infection.
Well, you know, I look at this world as in 2 parallel tracks. One track is we have the easy to induce immune responses, which is what came up in RV144, and I think all the animal model data and the clinical trial data suggest that those effector mechanisms are weaker than if we had broad neutralizing antibodies, which we've now spent 10 or 15 years learning that these are pretty hard to induce for a variety of reasons. And, but I agree with Nicole that we shouldn't neglect those weak responses because we don't have a really good challenge model. And we did see this protective effect. And we're actually starting to see a number of protective effects from a number of investigators with a number of non-human primate trials with really pretty good tier 2, tough to neutralize SHIV challenges. And we definitely not only should not ignore that, but we should have parallel tracks because we've got, I think it's a valid approach to try to make RV144 better. And one way to make RV144 better is all the work that was done suggested that it was this ADCC response targeted at particular regions of the virus. And so it worked in Thailand, and now can it work elsewhere? And that's the trial that Nicole mentioned. And can we broaden that response with easy-to-induce antibodies, for example, that have multiple, admittedly weaker protective mechanisms than broad neutralizing antibodies, but that together might get that protective efficacy above 50, 60% for a deployable vaccine. Yeah. So I think both tracks should continue to be worked on very hard.
And it's not just RV144 where we're seeing this correlation with things that are not broadly neutralizing antibodies. I mean, there are many other examples now. I think what it did was reawakened us, having spent my whole career in HIV working on broadly neutralizing antibodies, it's reawakened us to the other to the other end of the antibody, which I think is not a bad thing.
Well, we heard during this meeting, we heard Georgia Tomaris and I think a few other people talking about different types of antibody functions, like neutrophils mediated. I mean, there are several other antibody responses that seem to have a great role in protection, or at least in antibody responses.
And part of that, I think, will develop as we get more and more products into people and start getting a better handle on what correlates of protection really are. And I'm hopeful that all these new trials that will be starting in the next couple of years are going to fundamentally change our understanding of what we need.
You pointed out several times that we don't have a good challenge model. Mm-hmm. We heard George Shaw saying that now he has the SHIV model, the SHIV that we need. This is one, right? Do you think this could be a way to proceed?
Well, I think this is you correctly point out, this is a really important goal. And George's model is powerful because it's, number one, he figured out how to make SHIVs with virtually any virus envelope. And so we're no longer just stuck with what we get and on a haphazard kind of basis. So these are really designer SHIVs. And you can use the transmitted founder arms, which we know are what are important in starting off infections. And then next is how we use those. And I think that devising systems that more mimic natural transmission with very low dose challenges over long periods of time, which are very, very expensive, but are going to be a much better mirror of what happens in natural infections. And I think we can get much closer to it with much more useful challenges with these types of new chimeric viruses. So I'm pretty optimistic.
Yeah, you said it, and it's also very expensive because nowadays, if you have to do a low-dose multiple challenge, they're speaking about 8, 9, 10.
I'm actually thinking of more.
Yes.
More challenges. More challenges.
Definitely double digit.
So
yes.
It's getting very complicated, but possibly that's the model really to follow,
yes.
and then to use these new SHIVs or different SHIVs. But what do you think about SIV model for vaccine?
Well, I guess it also depends. I mean, the SHIVs are obviously perfect to look at antibody responses to HIV, which is really, if you have the immunogen that is supposed to induce antibody responses to HIV, the SHIV models are ideal. If you are thinking about anything else, and admittedly there are not that many things that are currently talking about anything else, but say Lewis's Pickers CMV, right? Clearly independent of antibody responses. Their SIV might still be a better model, even though SIV really is quite different from HIV. So again, I think you need to know what your limitations are in the models, and you need to tailor the model to what you're actually asking, or the question you're asking. So I think that there's use for both. I think it's really exciting to have these new SHIVs, but I wouldn't necessarily dismiss the SIV model. Yes, possibly for antibody responses, but definitely not for everything else.
You mentioned Lewis Picker's discoveries, and indeed, this is with attenuated cytomegalovirus as a vector to carry immunogens, and he's inducing no antibodies, but he's inducing CD8 cells that are not preventing infection, but are clearing the first round of infection. This is a big deal, I think, conceptually, regardless of, and I agree with what you say about the SHIV model, SIV models, but what he found was that the way the CD8 cells kill is in an atypical manner, and that they're killing by recognizing antigen presented in the context of this molecule HLA-E, which is relatively invariant in both humans and rhesus macaques. And secondly, that he's seeing antigen in the context of class 2, which traditionally is how CD4 cells see antigen. And the notion is that what may be protective is that the virus hadn't seen this type of immune response before. It's rarely, occasionally seen in TB and infection and vaccinations and such. And so exploring this new immunology, and it gets back to the point of really understanding the host virus interactions. Lewis may have found a pathway, a new pathway to harness in order to be able to fool the virus, if you will, and to prevent it from escaping, and to have more effective CD8 responses. So this would fall under one of those recent observations that are really potentially very transformative. And if this can be, so there's a lot of work going on right now to try to frame shift this into the human system and see, well, does it work in humans? But more importantly, what else could be inducing these kinds of atypical CD8 killing responses? And this, of course, is critical for the cure AIDS field as well.
Yeah.
So there are a lot of people excited about that.
Therapeutic vaccination.
Right.
Also, that's
Sure.
Yes.
something that we should consider.
Absolutely. I think that the cure field is very much in tune with considering the HIV vaccines as a big part of a potential cure. I think that the approaches that they've taken so far, which was only to drive out the virus from the reservoir, are clearly not working. So combination strategies might really be helpful there. And I think vaccines or therapeutic vaccines are a big part of that.
That brings me also to the, we heard a few talks on antibodies that are expressed in vectors, like so it's not administrating antibodies, broadly neutralizing antibodies, but to vectors that will produce antibodies. This is what is there are a few talks have been. Farzan is using such a model. Also, Mario Roederer spoke about it.
Yeah.
What do you think about that? Is that something we should proceed for and go for?
I think it's very helpful for increasing our understanding of what these neutralizing antibodies can do for preventing infection. I think it's even more important for considering them for cure research, honestly. This is the first time I heard a talk from Mario, actually, where they did do the AAV study and they did not start seeing antibodies against the insert.
Yeah.
Well, the insert, basically it is an insert, right? So previous studies that had been presented last year, actually, at Keystone, suggested that the monkeys will actually mount an immune response against the antibody, and then who knows what's going to happen. Now, all of a sudden, you have autoimmunity that can't necessarily be a good thing, right? So I think that it's a very important approach to gain more knowledge. I'm not sure I would be willing to be injected with AAV as a prevention for HIV necessarily, because I think that one of the big things that's currently under discussion is you basically make this antibody for the rest of your life. That's the idea, and that's what we're really aiming for. But at the same time, what if your immune system does something to this antibody where you need to shut it off, and that off switch is currently not available? So there's, I think, a huge amount of research going into that. I think that this is I believe something that at least is not relevant only for HIV vaccines, right? I mean, and there's a lot of gene therapy, so I think there are people outside of our field that are working on this as well, so hopefully there will be some synergy there. But it's really interesting to just see what these antibodies are capable of and the amount of reduction of viral load you can see in infected individuals. And again, the bar, sometimes you feel that the bar might be somehow different for therapeutic approaches versus preventive approaches, also just because the numbers are going to be different. The numbers of infected people are so much lower than the number of people at risk, right? So
I think we also mustn't underestimate the virus and its ability to escape from these antibodies. And in the context of ongoing viral replication and co-expression of whichever broadly neutralizing antibody you choose, I think the virus will win. So I think, no...
Well, don't say it like
triple
this.
combinations work for therapy, so triple combinations should work for it.
I think that's the key point, is that
Yes.
I think we shouldn't be thinking about that in terms of an individual isolated modality.
Not.
Because,
yes.
in the individuals who are making these antibodies, certainly the virus is winning there. So I think all of these things, for me, the AAV presents huge opportunities for learning, but I'm not sure that I can see it as a long-term therapy at this stage.
But it would be ideal.
Now,
one shot and you have it.
Wouldn't that be great?
Right?
Now, but where the field, I think, is moving in the near term to practical terms is a combination of long-acting antiretroviral drugs and the engineering of these broad neutralizing antibodies to last a long time. Right now, they last about 3 months, but the goal is for them to last 6 months, 9 months, maybe even a year, to get with a combination of drugs and monoclonal antibodies a shot per year.
And that's
which is very
passively
practical.
administered.
Passively administered, exactly. Just like a flu shot.
Which I would be more comfortable with personally than
Right.
that you like more.
That's right. That's right.
Well,
That's
right now it's still rather complicated because we're starting this trial soon about passive administration, and the visits of these participants are just very, very long. And at this point, because we don't have a long-acting antibodies, it's going to be a visit every 2 months where you're going to be sitting in a room for an hour waiting for the antibody to go in. So again, I think it's a wonderful study. It'll teach us so much.
Huge.
It'll teach us, hopefully, hopefully that, yes, it has been worth putting all this effort into designing the immunogens to induce these things. But it's hard. It's hard just implementing it. And so that's, I don't know if one of your questions was going to go into what are we seeing these studies as being? Is this a proof of concept simply, or is this something where we might move forward in actually trying to implement this? And I have a hard time seeing that as being, the passive infusions as being implemented. I mean, if we get the half-life to be a year, then yes, we're talking, but...
Well, it feels like the early days with AZT.
Yep.
I can remember the early days with AZT and being so impressed by that there's anything that has an effect, but the realization that the effect is so evanescent and small and short-lived. But perhaps it's a beginning, but I think it'll have to move on to become a yearly kind of thing to be practical. That's what I hear you saying.
Yeah, or I don't know if anyone, I'm assuming people are talking about putting it into a ring, right? A vaginal ring where it constantly gets released, slow release, or other forms of interventions where you can have slow release over time. So I think there are ways of approaching it, and I think that the study that we have planned right now is going to be so important because it's hopefully going to teach us the level. The AMP trial, the antibody-mediated prevention trial, is a trial where we are passively infusing one of the broadly neutralizing antibodies, the CD4 binding site antibody VRC01. It's infused every 2 months, and it's going to be infused at 2 different doses. And so we're going to very closely follow the pharmacokinetics of the antibody as it declines over time. And the hope is that we are going to be able to, by frequent sampling, pinpoint time of infection in breakthrough infections, assuming that there will still be some breakthrough infections. Because, I mean, wouldn't it be wonderful if there just weren't any? But then being able to pinpoint the time of infection and then knowing what the concentration of the antibody in the plasma was at that point to identify what a protective level of antibody is. Because if it's huge, then again, vaccines that are trying to induce these antibodies by active immunization are going to have a much harder time than if you can still show that you actually only require relatively minimal levels if they're there constantly to actually prevent infection. And again, then these release, slow release modalities would be something that are much more feasible at that point. So I think that there are a lot of things that, is asked about things that are excited that happened recently, is like I'm much more excited or really excited about things that are about to happen in the field. And we just have to wait and see what happens to those.
Yeah. I think there's an enormous amount we're going to learn from those trials. I don't think anybody's ever imagining using VRC01 as a prevention tool in the future. I mean, we already have better versions of VRC01, which is the antibody that Nicole's talking about, that will go into people. But I think the amount that we'll learn from that trial is just going to be extraordinary. It's going to be paradigm shifting, I think. I hope.
Yes. As long as we saw some protection, like fingers crossed.
Which other things are going on just now that you know would give some exciting results besides this one?
Well, especially after George's talk, I'm so excited about Bart's trial that is about to, well, it's not about to start, but it's going to start relatively soon, right?
Right. So we're testing the hypothesis that Penny brought up. We found in coevolution studies envelopes that can bind to the unmutated common ancestor of broad neutralizing antibody lineages, and we've worked to get these made by good manufacturing practice so that we can put them into humans and see if they start off the lineages in humans. And we have data in non-human primates and in humanized mice that at least the lineages can get started. And so this is be the first test of a concept of sequential immunizations to try to, just what Penny was saying, coax the lineages along and begin to see what it does against the human immune system.
When does it start?
Well, the first part of the trial will be in 2 parts. The first will be a dosing study of just the transmitted founder envelope alone, and that should start in June of 2016. And then early 2017, the sequential immunizations will start with the HVTN.
And how many immunogens are you talking?
Talking about 4 right now. It's a 4-valent,
Wow.
4-valent, and which may or may not be enough. Right. We'll see.
Yeah.
It was certainly enough to get made good manufacturing practice. So GMP. So we'll see.
Yes.
It will be a key challenge with these kinds of concepts that we're talking about. This idea that you can coax the immune system. It's going to have to be a balance between being able to coax the immune system and actually being able to administer these vaccines in a a real setting, and I think that will be a key challenge. But that's some time away.
Exact, and that's a good point, because we can't be too hopeful in the sense that we're making a product. We're really doing experimental medicine trials to learn proof of concept. And then once we find the enabling, we're still looking for the enabling technology. And once we find the enabling technology, then it becomes a technical problem of how to make it logistically doable and practical and implementable for in the field for whole cohorts of populations.
Yeah.
But that has also implication. If we go on the field with a product, a vaccine product, we know that we have to provide the population that will be treated with this vaccine, potentially immunogen, also with a whole array of other preventive means.
Yes.
Yes, everything is, and nothing happens in isolation, right? And so other advances in HIV prevention that are non-vaccine related are definitely very much influencing our work.
Yeah, that means that we have to provide PrEP, circumcision.
It's a good problem to have.
Yes.
But it is a problem, absolutely.
Yeah.
It will be something that we have to think about for the future.
Oh, no,
already.
we are already thinking about it. HVTN trials, especially right now with these larger trials in Southern Africa, it's very, very important to take into account everything else that's going on. So we couldn't do it just, again, in isolation. And it takes some time up front, really, to even define the trial itself, because if you start thinking about implementation of PrEP, or whether it's the new vaginal ring that has just shown some promise in a study.
Can you go a little bit more for our
I
audience?
don't. I unfortunately don't know very much about it. It was just recently presented at CROI and I wasn't there, but it is a ring that contains, I believe, an integrase inhibitor. Is that correct?
It's
an HIV treatment drug. Yes, I think it's the pivarine. But it basically showed that it's a ring that is similar to contraceptive rings that women use. And so it's basically dosing the drug, and it's been shown that if you are actually using the ring the way you're supposed to use it, and it's a 1 monthly ring, so basically each ring contains a dose that lasts you for a month. That if you continually use it, you can actually reduce infection by quite a bit. Unfortunately, as all of the other prevention modalities, it was highly dependent on the adherence of the women in the study. And there was a big gap in especially age, I think was one of the major determining factors, where the younger women were just not as willing or able to adhere to the regimen. But there are several modalities out there right now that are especially empowering women that are able to prevent infection. And while currently, at least, we don't make them available directly, we do absolutely facilitate access to these prevention modalities, whether that's PrEP or these rings or whatever is coming out of for any phase trial, whether starting at phase 1, the first time you go into humans. We absolutely provide standard of care, and then we provide access to anything beyond that.
Okay. You are involved in such trials too, in Africa.
I'm not involved in such trials, but I guess the point that you're making is that the size of these trials is now going to incrementally increase every time we successfully come up with a new way of protecting people from HIV. It's going to make the trials harder and harder to do, which is a challenge that we'll have to build into these kinds of...
Right, and so the calculations go in up front, and how your sample size calculations, basically, what your incidence rates are going to be expected.
It's one of the big challenges that we as HIV vaccine researchers face is why is what you're doing so important when this is a totally preventable disease with interventions that we already have available today if we could only implement them? And the answer, my answer at least is, and I'd be interested in yours, is that vaccines traditionally have been central and instrumental in eliminating epidemics. And if we had one today and could implement it, I have no doubt that it would play the same role for the HIV epidemic. And I view an effective HIV as a laudable goal in the setting of 2016, because it will be only one part of a whole portfolio of preventive measures, some of which may work for some folks and some of which may work for others, but together will end this epidemic. And the epidemic is not ended yet. And so we need to fill in this missing piece. And so I think it is a very important goal in the overall prevention portfolio.
Yeah, because the RV144 had already engaged
16,000.
16,000.
Yes, their incidence was a lot lower than they had originally calculated. So the numbers of infections were thankfully relatively low, but yes. Currently in South Africa, the numbers of participants that we're planning on enrolling are probably mostly between 5,000 and 7,000. So not quite the size of RV144, but reasonably large.
Not small trials, yeah.
That's enormous anyhow.
And these are phase 2B, not phase 3 necessarily, so some of them at least.
But for the new products that will come up in the recently now, we will have essentially more experimental medical trials, or small with that goes for new projects. Yeah, immunogenicity trials.
Safety trials.
Sorry.
Yes.
Safety trials. But first you have to see if your product is working, at least in small experimental medical trials. You were talking about that before, right?
Yes. And a critical part of that that Nicole is dealing with right now is to bring the technologies that Penny has in her basic science laboratory and I have in my basic science laboratory to the clinic. Because the way the HV10 and other organizations that carry out vaccine trials do the trials, you have to have what's called validated assays. And those are not the laboratory assays that I have in my lab and in other basic science labs, but yet it's the basic science labs that have the assays that need to be transferred into the clinical trial setting in order to understand the complex immunobiology and the host virus interactions in the context of vaccinations that we need to know whether it worked or not. Yeah. It's no longer just looking in the plasma and using only validated assays. So this is a big challenge. Maybe you could tell us just some of the hurdles you're trying to overcome, because it's a big deal.
Well, it's a big deal, especially because some of these assays are rather complicated, to say the least, which makes them so hard to validate, right? So I think that, yes, we do have our core validated assays, and thankfully the NIH hasn't pushed us too hard in validating every single last assay that we use. So I think for vaccine take, we will still use those, but we are fully aware that we won't be able to apply the same standards to all of the assays that are necessarily to really describe the immune responses induced by these vaccines to make sure that they are doing what we're expecting them to do. So there'll be a lot of work coming our way, but hopefully not quite as much work as validating every single last of these.
But I think you already have a precedent for that. I mean, with RV144, the pace with which you all moved in adapting existing assays from across multiple labs, I think has been a game changer in how you can combine these highly validated assays, which everybody understands are important, with what I think you're referring to,
which
more
is
kind of exploratory assays. And I think that changed the way we think about measuring vaccine efficacy and immunogenicity, and hopefully that will help the field.
Yes.
It's not easy to transfer everything from one lab to the other, and also on the field, because that's what is happening if clinical trials or vaccine trials are in Africa, in Asia. You have really to set up the logistics, implement laboratories, and what has happened in South Africa is extremely, it's wonderful.
An example of learning to do that, exactly that. Yeah.
So you have any other ideas of what is really stringent and something that you would like to...
I'm wondering what Penny's favorite trial or future thing happening is.
About your T cell, because we spoke a lot about antibodies. Antibodies all over. What about T cells?
Well, we mentioned exactly. We mentioned right now, really looking at all the trials that are either already happening or in planning, at least within the HV10. I think Lewis's is really the one that stands out as the one that at least is really aiming for T cell responses. I think the field really did turn after the STEP trial, where we tested a T cell based vaccine that was really just designed to induce T-cell responses and that unfortunately failed in showing any protective efficacy. So I'm very happy to work with the antibody people. I'm very excited about Lewis's study. And again, as I mentioned in the very beginning, I think that antibodies don't happen out of nowhere. Antibodies need B cells that we're looking at. Antibodies need CD4 T cell help. Antibodies need especially TFH cells. So learning how to design these immunogens to not only engage with the B cells, but also drive the right kind of help is going to be very important. And so I don't think that T cells are dead. I think that CD8 T cells are kind of hanging off the cliff a little bit. But at least for CD4 T cells, there's hope, and there's lots of assays that we're doing that are going to help understand really how these antibodies develop and what kind of help is needed to drive the immune response to where we need it to be.
I think that everybody nowadays thinks that we need these prime and boost approaches where we prime for T cells and then...
And I mean, we've seen that in many approaches where DNA primes are usually the ones that are really helpful for T cell responses. They don't really do much for B cells, at least not by themselves. But these combination strategies are really, really important. And I think that finding products that really help both the B cells and the T cells are going to be hugely important. And unfortunately, what we see right now is mostly that there is a divergence. So most prime boost strategies are going to be either really improving on T cells or really improving on the antibodies, but we haven't really found anything that does both at the same time. But there are so many more combinations out there that we can try, right? Even with existing products. So I think that those smaller trials are really going to be leading the way in trying all these different things in a small number of people so they're not that expensive, because that's really what's driving everything, right? It's the cost of the trials. So having smaller trials that are going to be able to be done more quickly will teach us a lot, and hopefully we kind of pave the way of making those happen.
So thank you very much. It was for me, it was a very enjoyable discussion. I hope it was for you too. And I hope it's also for our audience that it will be helpful for everybody. And I thank, on behalf of the Global AIDS Vaccine Enterprise and of the Keystone Symposia, that you have been joining this panel discussion and have been really discussing heavily.
And enjoying. It was a pleasure.
Thank you very much.
Thank you.